Welcome back to the deep dive. Today, we're really trying to crack the code of, well, cancer communication. Imagine the tumor microenvironment, right? Not just a battlefield, but maybe more like a noisy, chaotic digital network. And the immune system is in there trying desperately to coordinate a defense against this invader that almost speaks its own language.
Exactly. And that language, those essential signals like text messages or urgent emails flying back and forth, is carried by proteins we call cytokines. So today, we're diving deep into some recent research on how these factors really control a tumor's fate. The key thing, I think, we need to grasp is their dual nature. Some cytokines, they lead the anti-cancer charge. Others, though, actively fuel the disease's growth.
Okay, so that's our mission then. Understand that contradiction, that sort of biological push-pull, and maybe more importantly, why has targeting these pathways therapeutically been so incredibly difficult for so long?
All right, let's start with the basics, the core definition for you. Cytokines are, at their heart, cell-to-cell communication factors, usually small proteins. Immune cells primarily use them to coordinate a massive response. Now, characteristically, they're generally labile, meaning they're fragile, they break down quickly, and they typically act locally. So, signaling back to the cell that made them, that's autocrine, or just to cells nearby, that's paracrine. Only rarely do they act systemically over long distances, like signals heading to the liver or maybe the central nervous system.
All right, so here's maybe the concept that explains why this whole field is just so complicated. This idea of pleiotropy. You're saying one specific cytokine, let's take IL-6 for example, can have totally different effects depending on which cells happen to be around and have the right receptor.
That's exactly it. The final biological outcome is entirely dependent on which specific cell types are present and expressing that required cognate receptor. Think of it like sending the exact same memo to, say, a dozen different departments in a company. Each one acts on it differently based on their specific function and context.
That makes sense. And that contextual dependence really explains why the established tumor microenvironment, the TME, is such a complex mess to deal with. So while we know there are adaptive heroes, like IFN gamma-fighting tumors, we first need to talk about the dark side, the innate inflammatory cytokines that actually help cancer get started.
Yes, we really have to address that first. Let's start with those enemies within. The key players we should focus on here are IL-1 beta, TNF-alpha, and IL-6.
Okay, IL-1 beta, TNF-alpha, IL-6. Got it.
These are mostly made by myeloid cells, the immune system's first responders. Their main job initially is to kick off inflammation, make blood vessels leaky, call in more immune cells. But here's the twist. When that inflammation becomes chronic, their role changes. They can actually start giving direct growth and survival signals to early cancer cells, often using pathways involving transcription factors like NF-kappa B or STAT3. This provides that molecular link people have suspected for ages between chronic inflammation and actually developing cancer.
The story around IL-1 beta seems like a perfect case study of this whole dilemma. Didn't some studies on lung cancer show it could actually boost tumor growth?
Yes, exactly. Research suggested that IL-1 beta, produced by those tumor-associated macrophages, could indeed potentiate oncogenesis, basically feeding those early tumor cells, especially in response to things like air pollution particles. And that finding was a big driver behind the CANTOS trial, which was quite landmark. The initial promise was enormous. They used an antibody, canakinumab, to block IL-1 beta. And in patients who already had high inflammation levels, it led to a really significant, like 70% reduction in the risk of developing lung cancer later on. So it looked like IL-1 beta blockade could be a star player, especially for prevention or interception.
Okay, but that's the confusing part. If it worked so well for interception, why did it seem to, well, flop once cancer was already established?
That's the crux of it. When they ran large trials testing IL-1 beta blockade with chemotherapy or checkpoint inhibitors in patients who already had established solid tumors, well, they just failed to show a benefit. It seems the complexity and, importantly, the redundancy within that established TME just overwhelmed the effect of removing that single factor. The system found workarounds.
So the tumor microenvironment is just too robust by then. What about its partners, IL-6? I know it has systemic effects like telling the liver to make certain proteins during inflammation, but what about inside the tumor?
Right, in the TME, IL-6 often signals through its partner protein, GP130, and this promotes tumor growth mainly via activating STAT3. And just like IL-1 beta, blocking IL-6 largely failed to shrink established tumors in trials. But, and this is a critical but, we can't just dismiss IL-6. blocking it is absolutely crucial clinically for managing cytokine release syndrome, or CRS. That's that potentially deadly side effect you can get with CAR T-cell therapy. Blocking IL-6 helps dampen that dangerous inflammatory storm, so it has a vital niche role.
And TNF-alpha, that one seems even more contradictory. I feel like I've heard it associated with both killing cancer cells and helping them grow.
You have. It's a tricky one. It can induce what's called extrinsic apoptosis, basically, tell some tumor cells to self-destruct, which is good, right? In fact, a recombinant form of TNF-alpha was actually approved for treating sarcomas, but only when delivered in a very specific way, isolated limb perfusion precisely to avoid overwhelming systemic toxicity. The big downside, though, is that TNF-alpha is also a major driver of those immune-related adverse events, the IRAEs, that can happen with checkpoint blockade immunotherapies. So blocking TNF-alpha is often an effective treatment for side effects like immunotherapy-induced colitis. But at the same time, the cytokine itself can act as a growth factor for certain cancer cells. It really is a classic double-edged sword.
Okay, so that pattern of failure blocking just one cytokine doesn't defeat established cancer. It really tells us the underlying communication network is just too resilient, too complex. If we want to design better therapies, we need to get deeper into the mechanics. How are these signals actually processed inside the cell? Let's get into the plumbing.
Absolutely. The main signaling highway for most of these cytokines is the JAK-STAT pathway. Now think about this. We have over 100 different cytokines. But in humans, there are only 4 types of Janus kinases, or JAKs, and only 7 types of STAT proteins. Those are the signal transducers and activators of transcription. So what that suggests is the specificity isn't so much about the final command sent to the nucleus. It's more about which cell sends the signal and which cell receives it. The context matters more than the core machinery.
Okay, let's visualize this JAK-STAT process, like an activation chain reaction.
It is, and it's remarkably fast. So first step. The cytokine, the ligand, binds to its specific receptor on the outside of the cell. This binding forces the different parts of the receptor protein to physically come together. That's receptor dimerization. Imagine it like turning a key in a lock that snaps 2 pieces of machinery together. This proximity then causes the JAK proteins, which are hanging out nearby inside the cell attached to the receptor tails, to activate each other. They essentially slap a phosphate group onto their partner J, that's JAK cross-phosphorylation.
Okay, so now the JAKs are switched on, their active enzymes. What do they target next?
Their next target is the receptor itself. They phosphorylate specific spots on the receptor tails, creating docking sites. These docking sites are perfectly shaped to grab onto STAT proteins, which contain a specific region called an SH2 domain that recognizes these phosphorylated sites. So the STATs bind, the activated JAKs then phosphorylate the STATs. Once phosphorylated, 2 STAT molecules pair up, they dimerize, and the STAT dimer then quickly moves into the cell nucleus. And once it's in the nucleus, it binds to specific DNA sequences and activates gene transcription, turning on the cell's response program.
Wow, that sounds like a very rapid on switch, which means there must be equally rapid off switches, right? Otherwise, the cell would just be stuck in an on state. How does the body naturally regulate this?
Absolutely crucial. Yes, the system relies heavily on negative feedback loops. The first big one involves proteins, called SOCS proteins. This stands for suppressor of cytokine signaling. They work in a couple of ways. They can bind directly to the activated JAKs and physically block their activity, that's steric hindrance. And really importantly, the STATs themselves actually turn on the production of SOCS proteins. So the activation signal literally creates its own inhibitor. It's a beautiful self-limiting loop. SOCS proteins also recruit other cellular machinery to tag the JAKs for destruction, basically degrading the signaling components.
Right, and that's why understanding SOCS is so critical for therapy, isn't it? If a tumor cell learns to ramp up SOCS production to shut down beneficial immune signals, well, then maybe blocking SOCS could be a way to treat the cancer.
Precisely. That's an active area of research. The second major mechanism involves enzymes called phosphatases, specifically ones like PTPN2 and PTPN1. These act like molecular erasers. They remove those activating phosphate groups from both the JAKs and the STATs, shutting down the signal. And yes, inhibitors targeting PTPN2 and PTPN1 are being developed. They've shown some real promise in mouse models for boosting anti-tumor immunity, essentially by letting the on signal last a bit longer.
Okay, quickly though, not every cytokine uses JAK-STAT, right? What are the other major signaling families we should know about?
Good point. 2 other really crucial families that use different mechanisms are the transforming growth factor beta, TGF-beta family, and the TNF superfamily. TGF-beta signaling involves a different set of intracellular proteins called SMADs. When TGF-beta binds, the receptors rotate, phosphorylate SMADs, which then partner up and head to the nucleus. The TNF superfamily, which includes TNF-alpha, as we discussed, is also different. Their receptors are trimers, binding trimeric ligands. Depending on the specific receptor and cell type, they can either activate that NF-kappa B pathway, which is common in immune cells and many cancers, or they can trigger extrinsic apoptosis cell death through components called death domains. Also, the IL-1 family receptors use TIR domains and recruit MyD88, activating NF-kappa B, similar to toll-like receptors. They also have that interesting activation step involving inactive precursors cleaved by caspase-1. Understanding these parallel signaling highways really highlights why blocking just one cytokine pathway often fails the tumor, can often just reroute growth signals through another available lane.
Okay, let's pivot now. We've talked about the problematic ones. Let's talk about the champions of adaptive immunity, starting with the real star player. Interferon gamma, IFN gamma.
IFN gamma. Yes, definitely the MVP in many ways. It's produced mainly by activated CD4 plus T helper cells, CD8 plus killer T cells, and also natural killer, or NK, cells. It signals through the JAK-STAT pathway, like many others, and it's absolutely critical for the immune system to successfully eliminate tumors.
And the proof for its importance, the whole concept of immune surveillance, really came from those groundbreaking studies, wasn't it, by Robert Schreiber's group years ago, showing mice whose tumor cells couldn't respond to IFN gamma developed tumors much faster.
That's right. Those were landmark studies back in the 90s and early 2000s. They definitively showed that the immune system actively surveils and destroys nascent cancer cells, and that this process critically depends on IFN gamma signaling. Its anti-tumor effects are really pleiotropic. There are many. It can act directly on tumor cells to slow their growth or induce senescence, a state of irreversible growth arrest. But perhaps more importantly, for coordinating the immune attack, it forces tumor cells to display more antigens by upregulating MHC class I and class II molecules. It also boosts the machinery needed to process those antigens.
Which means that the success of our modern immunotherapies, like checkpoint inhibitors, really hinges on this cytokine still working properly.
Absolutely. The efficacy of immune checkpoint blockade, ICB, is definitely dependent, at least in part, on functional IFN gamma signaling within the tumor. We often find that tumors developing resistance to ICB have acquired mutations that break some part of that IFN signaling pathway. They're essentially trying to become invisible again. But remember, there's always a trade-off. IFN gamma also causes cells, including tumor cells, to upregulate PD-L1. That's the very ligand that checkpoint inhibitors like anti-PD-1 are designed to block. PD-L1 acts as a brake on T cells, so IFN gamma simultaneously helps reveal the tumor and provides a mechanism for the tumor to defend itself. Duration of signaling also seems key. Prolonged exposure might favor pro-tumor effects. And unfortunately, just like its inflammatory cousins, trying to give recombinant IFN gamma systemically as a therapy generally fail due to toxicity and pretty poor efficacy.
Okay, so the same story, powerful anti-tumor effects locally, but problematic systemically seems to apply to IL-12 as well, another major pro-immunity cytokine.
Yes, IL-12 is incredibly potent. It's mainly produced by dendritic cells, the professional antigen presenters, and macrophages. Its superpower is strongly pushing CD4 plus T cells towards that Th1 phenotype, the type that becomes a major producer of IFN gamma. The advantage is that even tiny amounts of IL-12 delivered locally within the tumor can trigger a massive and sustained wave of IFN gamma production right where you need it.
But I recall the word disastrous being used in relation to early systemic IL-12 trial.
It was, unfortunately. The toxicity was just unacceptable. Giving IL-12 systemically creates such a widespread, overwhelming inflammatory response like a cytokine storm that patients simply couldn't tolerate it. The therapeutic window was essentially non-existent. And that is why so much current research focuses heavily on local delivery strategies, things like engineering viruses or even immune cells to produce IL-12 only after they've reached the tumor microenvironment. Keep the potent signal contained.
Let's move on to what you might call the T-cell lifelines, the crucial IL-2 family of cytokines, starting with IL-2 itself. Now, if IL-2 is so essential for T-cell survival and function, why isn't just giving patients more IL-2 the obvious answer?
Yeah.
Doesn't the immune system just need more fuel?
That's the IL-2 trap. You're right, activated T cells absolutely need IL-2 to survive and proliferate. They express the receptor components, including the high-affinity alpha chain, IL-2R alpha, also known as CD25, when they get activated, but, and this is the huge hurdle, so do regulatory T cells, or TREGs. Those are the FOXP3 positive cells whose job is normally to suppress immune responses and prevent autoimmunity. The problem is, TREGs constitutively express, meaning they always have on their surface that high-affinity IL-2 receptor alpha chain CD25.
Ah, okay. So in the tumor microenvironment where IL-2 might already be scarce, the TREGs effectively act as IL-2 sinks. They just soak it all up.
Precisely. They outcompete the killer T cells, the CD8 plus T cells we want to activate, for the limited IL-2 supply. They just gobble it up because they have more of that high-affinity receptor. This was a major reason why the historical attempts to use high-dose recombinant IL-2 therapy were so problematic. Yes, it was approved for melanoma and kidney cancer, but it caused severe systemic toxicity, most notably something called vascular leak syndrome, where fluid leaks out of blood vessels. And ultimately, its efficacy was limited because it often ended up preferentially expanding these immunosuppressive TREGs just as much, if not more, than the effector T cells.
Okay, that explains the failure of simple IL-2. So that brings us to what some people call the better brothers, IL-15 and IL-21. What makes them different?
Right, IL-15 is really critical for the function and persistence of both NK cells and CD8 plus T cells, especially memory T cells. It actually shares some receptor components with IL-2, like the beta chain. But here's the key difference. Stimulating cells with IL-15 promotes the proliferation of these important effector cells, NK and CD8 plus T cells, but it does not expand TREGs. That's a huge advantage. Furthermore, IL-15 often works through a cool mechanism called transpresentation. Dendritic cells or macrophages will bind IL-15 to its own specific alpha receptor chain, IL-15R alpha, on their surface, and then physically present it to the NK or T cell. It's like a direct handoff. And IL-21 is similar in that it supports the proliferation and cytotoxic function of NK cells and CD8 plus T cells, again, without significantly expanding those suppressive TREGs. So these offer ways to boost the good guys without boosting the bad guys.
Got it. Before we move to the final piece, chemokines, let's quickly touch on those other growth factors that tumors often co-opt. You mentioned GCSF, MCSF, and GM-CSF. What's their role?
Right, those are colony-stimulating factors. GCSF stimulates granulocyte production, MCSF stimulates macrophage production, and GM-CSF stimulates both. Their main job is signaling the bone marrow to replenish these types of myeloid cells. Now, tumors are clever. They co-opt these signals. For example, many tumors produce MCSF to attract and maintain a population of tumor-associated macrophages, or TAMs, which are often immunosuppressive. However, we've also learned how to harness some of them. GM-CSF, for instance, was actually a key component in early cancer vaccine strategies like GVX. Why? Because it helps recruit and activate beneficial dendritic cells, the ones needed to kickstart a T-cell response. It's also engineered into the oncolytic virus TVEC for the same reason. So again, context is everything.
Okay, finally, let's talk about the navigators, the chemokines. These are specialized cytokines, right? Their main job is directing traffic, telling immune cells where to go.
Exactly. They are the trafficking signals. They form chemical gradients within tissues, often by binding to sugars on the extracellular matrix called JAGs, glycosaminoglycans. Immune cells follow these gradients to find their way. And just like with the other cytokines, we see clear examples of chemokines driving good traffic and others driving bad traffic into the tumor.
Okay, give us the good traffic first.
The good traffic is exemplified by the CXCR3 axis. Activated T cells, the ones we want in the tumor, express the receptor CXCR3. This receptor binds to chemokines like CXCL9, CXCL10, and CXCL11, which are often produced in response to, guess what, IFN-gamma. So you get this positive feedback loop. IFN-gamma is produced, it induces these chemokines, which then recruit more CXCR3 positive T cells, which can make more IFN-gamma. This axis is actually known to be really important for the success of anti-PD-1 therapy.
Makes sense. Feed-forward loop.
Yeah.
And the bad traffic.
That's often associated with the chemokine CCL2, which attracts monocytes expressing the receptor CCR2. These monocytes often differentiate into those immunosuppressive TAMs we mentioned earlier. High CCL2 often correlates with poor outcomes. Now you might think, okay, let's just block CCL2 or CCR2, but attempts to do that therapeutically have been tricky. Monocytes are critical for normal host defense, so blocking them broadly can be problematic. Plus, the immune system often has redundant pathways. If you block one route for suppressive cells, sometimes another chemokine like CXCL8, also known as IL-8, might ramp up and recruit neutrophils instead, which can also support tumor growth and angiogenesis. CXCL8 itself is often induced by those pro-inflammatory cytokines like IL-1 beta and TNF-alpha we started with.
So we've kind of circled right back to where we started, haven't we? This blockade paradox. We have fantastic targeted drugs, monoclonal antibodies for many of these cytokines. But blocking just one has largely failed to cure established solid tumors. Why does it seem easier, relatively speaking, to dampen down chronic inflammation in an autoimmune disease than it is to successfully initiate and sustain anti-tumor immunity in cancer?
It's a fundamental challenge. It boils down to a few things. One is Likely the narrow therapeutic window we talked about, the dose that helps, might be very close to the dose that harms systemically. Another is the sheer redundancy and complexity of the established TME. Autoimmunity often involves turning off an overactive but relatively defined response. Cancer immunotherapy requires turning on a response, often against a target that looks quite similar to self, directing it specifically, sustaining it for potentially months or years, and doing all that without causing fatal autoimmunity or cytokine storms. It's fundamentally harder, perhaps, to initiate and orchestrate a complex attack than it is to just dampen down an existing fire. The natural system is just incredibly complex and robust against single-point interventions once cancer is established.
But the story doesn't end there, right? The future isn't just about giving up on cytokines. You're saying the solution lies in smarter design and, crucially, localized delivery.
Absolutely. That's where the real breakthroughs are happening now. We are seeing success, but it's coming from being much smarter about it. Look at the local delivery methods. For example, an engineered molecule combining IL-15 with its receptor alpha chain, IL-15R alpha, fused to an antibody Fc domain for stability. That drug is now FDA approved. But importantly, it's approved for intravesical treatment, meaning delivered directly into the bladder for patients with BCG-unresponsive bladder cancer. The key lessons are right there. 1. Local delivery avoids systemic toxicity. 2. Stability matters, that Fc fusion extends the drug's half-life dramatically compared to natural IL-15. And 3, you're often targeting patients who already have some level of pre-existing anti-tumor immunity that just needs a boost. You're augmenting, not starting from scratch in a completely cold tumor.
So we're really entering an age of sophisticated protein engineering, designing cytokines specifically to overcome their natural limitations.
Exactly. The creativity is amazing. Researchers are designing mutated versions of IL-2 that have been tweaked so they don't bind well to that high-affinity IL-2R alpha chain found on TREGs, preferentially stimulating the effector cells. Decoy-resistant cytokines. For instance, IL-18 is another pro-inflammatory cytokine, but the body makes a natural inhibitor called IL-18 binding protein, IL-18BP. So people are engineering IL-18 variants that are potent but resistant to being neutralized by IL-18BP. Even pro-cytokines or fusion proteins where the cytokine is inactive until it reaches the tumor microenvironment and gets activated specifically by conditions found there, like lower pH or tumor-specific enzymes called proteases.
That sounds less like just hitting the gas and more like, actually programming the immune response with incredible precision. So despite all the historical difficulty, the failures of systemic therapy, these recent successes with engineered variants and localized delivery, they really show us where the field is heading, don't they?
I think so. Which leaves us, and you listening, with this final, maybe provocative thought to consider. If we really can design a cytokine, or a cytokine delivery system, to activate only the precise immune cells we need, only when and exactly where we need them within the body, could the next true revolution in cancer treatment be less about just immune blockade, taking the brakes off the system, and more about highly specific immune programming, giving the immune system the directed, surgical, potent fuel it needs to finally win the fight?
Something to definitely think about. Thank you for joining us on the Deep Dive.